hello from Martha's Vineyard, actually. Thank you, Dorothea and Jim and Elias for this opportunity to speak here. So my group has been working to understand the molecular mechanisms that underlie membrane maintenance and growth in organelle biogenesis, and I'm delighted to share our thoughts as to how these processes could happen. And what you see here is a schematic of a cell's various organelles, each surrounded by a lipid layer. The membranes surrounding each of these organelles are different in their bulk lipid composition and in their composition of signaling lipids. And for cells to live, they have to maintain these distinct organellar membrane compositions with their distinct biochemical and biophysical properties. And yet, most of the lipids that form cellular membranes are synthesized primarily in the endoplasmic reticulum and then redistributed to other compartments from there. It was long thought that bulk lipids, like proteins, move between organellar membranes by vesicle trafficking. But it's also been clear that there must be additional mechanisms for lipid transfer. So this is because lipid transfer by vesicle trafficking would tend to equilibrate lipids between different organelles, ablating the asymmetry that is so important. This is because vesicular transport is relatively slow, and there have to be means to replenish lipids rapidly. And then this is because there must be means to transfer lipids to organelles that are disconnected from trafficking pathways, like peroxisomes or like mitochondria. So we now appreciate the importance of non-vesicular protein-mediated lipid transport at so-called membrane contact sites, where organelles are closely opposed, often between the ER, where the lipids are synthesized, and another organelle. And transport at these sites can explain how lipids can be moved so quickly and selectively and supplied to organelles that are disconnected from the secretory pathways, such as mitochondria. Until maybe 5 years ago, eukaryotic lipid transporters were all thought to comprise teacup-shaped modules with a hydrophobic cavity that can solubilize one or a small number of lipids. These modules can shuttle back and forth between organellar membranes, but we now think that most such shuttle-like proteins primarily exchange lipids between membranes, and so they're not well suited to function in net transfer. So that left open the question as to how bulk lipid transfer to non-secretory organelles is achieved. So today I'd like to tell you about another family of lipid transporter whose founding member is VPS13 that we discovered together with the groups of Pietro De Camilli and Tom Milia in our Department of Cell Biology at Yale. These are elongated proteins that can span between organelles at contact sites and that act as bridges for lipids to move between membranes, and so they're referred to as bridge-like lipid transport proteins, or BLTPs. These proteins are evolutionarily conserved from gram-negative bacteria to mammals, and this panel shows the distribution of known eukaryotic BLTPs at different contacts, where they play different roles. They are involved in a host of processes, including the expansion of new membranes during organelle biogenesis, for example, of the autophagosome, or for the maintenance or repair of other membranes, for example, the lysosome or mitochondria. Arguably, the best characterized BLTPs are those in the ATG2 and the VPS13 families. And our group has worked on both of these families, although recently we've been focusing more on the VPS13 proteins. These are of particular biomedical interest because their dysfunction is associated with severe neurological disease, including Parkinson's or VPS13C, which this audience may already be well acquainted with, and then chorea acanthocytosis or VPS13A. The different disease manifestations reflect on the different functions of these proteins at different contact sites. Our current understanding is that BLTPs do not work alone, but rather in complexes with partner proteins, and that BLTP function is regulated by their localization to their sites of action at specific contacts, and then the assembly of BLTP-containing lipid transfer machinery there. BLTP partners can include adapter proteins to anchor the ends of the BLTP bridge domain to the donor and the acceptor organelles, thus localizing a given BLTP to the correct contact site, as well as integral membrane proteins, which may facilitate the transfer of lipids at the BLTP membrane junction. Identification of partner proteins is ongoing, and it's clearly prerequisite for understanding the molecular mechanism that underlie BLTP-mediated lipid transfer and its regulation. So an emerging paradigm based primarily on studies of the VPS13 and the ATG2 proteins is that at the acceptor membrane, BLTPs cooperate with membrane-embedded scrumbases. So scrumbases equilibrate phospholipids between membrane leaflets, allowing for the concomitant expansion of both leaflets of the membrane, and so membrane growth, even as the BLTP is delivering lipids only to the cytosolic leaflet. That is, we are starting to understand that BLTP-mediated lipid transfer means transfer across the cytosol and between the leaflets of membranes. The mechanisms governing the transfer of the lipid from the bridge domain to the membrane and the role of the scrumblase in this process have been elusive in the absence of near-atomic resolution information regarding BLTP scrumblase complexes. So I've just shown you a cartoon that's suitable for a textbook insofar as that it glosses over so much that we do not know yet about how these proteins work. We don't know the identity of all the partner proteins in these complexes. We suspect that their activation involves correct localization, but we don't yet understand fully what localization entails. We don't know how lipids are passed between membranes and the protein bridges to allow for fast, efficient transport. And we don't know why lipids move directionally from the ER to other organelles, although everyone agrees that there is some driving force, which we will be arguing could be differences in membrane tension. So we've been focusing on VPS13 family proteins, in part for their disease relevance, but mainly for a very practical reason. Namely, unlike BLTPs and other families comprising the bridge domain that function with exogenous adapter proteins, the VPS13s have built-in adapter modules and so already represent partially assembled transport complexes. But I'll tell you two intertwined stories today. The first pertaining to the Parkinson's protein, VPS13C, which is not partnered with a membrane protein, and that represents how VPS13Cs and VPS13 look before they interact at contacts. And the second story is about VPS13A as it interacts with a membrane receptor, the scrumblase XK. The precise function of all the BLTPs, including VPS13C, are still under investigation, and the De Camilli group only recently succeeded in discovering a function for VPS13C at lysosomes, namely in the repair of damaged lysosome membranes, presumably in the transfer of phospholipids from the ER to the damaged membrane, and they started to work out the mechanism for its recruitment to damaged lysosomes. They found that VPS13C is mostly cytoplasmic, but it goes to ER lysosome contacts in response to lysosome damage, in this case induced by a drug called LOMI. They found that localization depends on the presence of the lysosomal marker, the RAB7 GTPase, and on a subset of VPS13C's built-in adapter modules. So here at the top, you can see a schematic showing VPS13C domain architecture. You can see that this is an enormous protein. The portions that correspond to the lipid transferring bridge domain are light blue, and then the built-in adapter modules at its C-terminal end are shown in violet, in lilac, and in shades of green. So based on cellular studies in the De Camilli group, the amphipathic helices at the C-terminal end called the ATG2 C motif, because they're also present in the ATG2 family of BLTPs, act as a sensor for damaged membrane. And then further, the so-called VAB adapter module of VPS13C is required for localization. Intriguingly, they found that the VAB domain alone goes to lysosomes constitutively, even while the intact VPS13C is usually cytoplasmic. And so the different localizations of intact VPS13C and the VAB domain suggested that lysosome damage somehow precipitates a conformational change in intact VPS13C, whereby a site that's required for binding becomes accessible. While quite a bit was known about the overall architecture of VPS13 from previous structural studies from our group, and also from the magic that is AlphaFold, we did not have an experimental structure for any of the intact VPS13s, showing the disposition of the various adapter modules. And so we undertook structural studies of intact VPS13C as a basis for better understanding the kinds of interactions involved in its recruitment to lysosomes. When I say we, this mainly refers to Daji Lee, a graduate student in our group who spearheaded this project. So here in the left panels, you see the composite map based on 3 cryo-EM maps for VPS13C that Daji calculated, with average resolutions ranging from 3.8 angstroms at the C terminus to 4.1 angstroms at the N terminus and 4.2 angstroms in between. The right panels show the model that Daji built into these maps. The bridge domain is again shown in light blue. You can see the VAB in light gray, and at lower resolutions because they're mobile, the pH domain in blue and a WWE domain in lilac. Note that both the pH domain and the WWE domain are closely associated with the bridge domain, and they're not detached. In contrast, the amphipathic helices in the ATG2C motif are mobile and most likely are unstructured in the absence of membrane. You also see dark density that does not correspond to VPS13C at the VPS13C N terminus. And in consultation with Xinbo Wang and Hongyan Hao in the Decamilli lab, Daji identified this density as corresponding to calmodulin. So the Decamilli group had found by a mass spec analysis that calmodulin, which is present at high levels endogenously in the XB293 cells, which we used for protein expression, co-purifies with VPS13C. And AlphaFold predicted that calmodulin should bind exactly where we see this gray density. Calmodulin has 2 lobes, each with 2 calcium-binding EF hands. Each of these lobes interacts with a segment in VPS13C that spans over the bridge domain. We confirmed that calmodulin co-purifies with VPS13C by Western blotting, and we confirmed its binding site by a mutational analysis that you can see here at the right. You can see that mutations in the putative calmodulin binding site abrogate the interaction with calmodulin. Although the expression levels of the mutants that no longer bind calmodulin are similar to wild-type protein, and the mutant proteins are soluble and they don't aggregate, they have a different profile by size exclusion chromatography and by negative stain EM, and they don't fold into rods, indicating probably a folding defect. So either the mutations cause the folding defect, or else VPS13C requires calmodulin for proper folding. Likely this latter scenario is true. Bacteria do not have endogenous calmodulin, and we can only make VPS13 fragments that include the calmodulin binding site in bacteria if we co-express them with calmodulin. Of course, the calmodulin binding site is at the very end terminus of VPS13, where VPS13 interacts with the ER. So we're in a cell biology department, and so we also tested whether the VPS13C calmodulin binding impairing mutations affect VPS13C localization. And what you can see here is that they do. Even though the mutant constructs are not aggregated in these images, given indications that the mutants don't fold from our negative stain characterization, we can't tell whether their mislocalization is due to the misfolding of VPS13 or because calmodulin is part of the machinery responsible for localization to the ER, or both. But I think it's safe to say that calmodulin binding is important for VPS13C localization. We also found that the human VPS13s that are known to localize to the ER and a fungal VPS13, which likely does, at least sometimes, they all bind calmodulin in the same way. as VPS13C. So we would propose calmodulin, or perhaps another member of the calmodulin superfamily, as an interactor for all of these VPS13s. Given that calmodulin binds near the N-terminus, where VPS13 associates with the ER membrane, and its role in localization, we think that calmodulin could have a role as an exogenous adapter for VPS13s at the ER. Moreover, the well-established role of calmodulin in cellular calcium signaling and the importance of contact sites in the regulation of calcium levels suggest that calcium signaling could play a role in VPS13 ER localization. But this could be due directly to cellular calcium levels, resulting in conformational changes in calmodulin and then its interaction with VPS13, or it could be indirect, for example, through phosphorylation by calmodulin-dependent kinases. We note that there are several phosphosites at the VPS13 N-terminus, including for calmodulin-dependent kinases, and also at the VPS13C C-terminus. So now let's go back to VPS13C architecture, and in particular, the lipid transport part of the molecule, the bridge domain. So as known from previous experimental structures and also AlphaFold, the bridge domain looks like a taco shell comprising a series of repeating beta groove motifs. There are 13 in the case of VPS13C. These are 5-stranded anti-parallel beta sheets that are arrayed end to end. And then there are several additional beta strands at either end that I'll refer, and also a few alpha helices at these ends that I'll refer to as N and C terminal caps, and that I'll talk more about shortly. The lipid transfer groove is lined with hydrophobic residues that can solubilize lipid fatty acyl moieties, whereas the hydrophilic lipid head groups would be exposed to cytosol. Due in part to the limited resolution of these reconstructions, and also because each reconstruction represents the average of hundreds of thousands of particles where lipids don't necessarily have well-defined binding sites, we can't resolve lipids in the channel, although we do see low contour density that likely is corresponding to lipids. But I promise that I'm going to show you some ordered lipids in a BLTP in the second part of this talk. The width of the transport groove that runs along the concave surface of the taco shell is variable in the experimental structure of VPS13C as analyzed in this panel here, ranging from about 10 angstroms across to 55 to 60 angstroms across. So experiment-based models have recently become available for several other BLTPs in addition to VPS13C, and also the fungal VPS13, as was also modeled now by Dachi Li, VPS13A by Bodan Hu in our group, and then for ATG2, and then a fragment of BLTP1. And the width of the lipid transfer channel is variable along the length of the bridge domain in all of these proteins. Whether this variability is physiologically significant is unclear, which will not stop me from speculating that one mechanism to regulate lipid transfer rates could be to regulate the width of this lipid transfer channel. Notably, the lipid transfer channel is wider in all of the VPS13 proteins compared to either ATG2 or BLTP1, and it's significantly wider in VPS13C as compared to VPS13A. So the bridge domains of VPS13A and C are shown at scale here, and you can also see a superposition of VPS13C and a fragment of the fungal VPS13. We posit that the width of the lipid transfer channel could impact lipid flux, so that the VPS13s and VPS13C in particular may be able to transfer lipids at faster rates than other narrower BLTPs. So now let's talk about the C-terminal end of VPS13C, and I'd like to draw your attention to the VAB adapter module in light gray. And what you should see is that it arches over the end of the bridge domain so that the bridge domain cannot access membrane. Now I'm going to rotate VPS13C in this left panel by 90 degrees, like so. So you can see now the VAB domain blocking the end of the bridge. You can also see that the end of the lipid transfer groove in the bridge domain appears to be blocked. And so VPS13C in solution is in a lipid transfer incompetent conformation and has to undergo conformational changes in order to function as a lipid bridge. In this inactive conformation, the VAB is buttressed via a helix at the very C-terminus of VPS13C. As I'd mentioned before, a possible regulatory role for calmodulin kinase phosphorylation, I should point out that there are two calmodulin kinase dependent phosphorylation sites at the very end of the helix, and that these could play a role in reconfiguring the VAB domain and making it and changing its orientation so that VPS13C becomes lipid transfer permissive. And here you can see that the VAB domain is stabilized in the lipid transfer incompetent conformation by linker elements from the C-terminal cap, shown in gold here. One such linker is snaking along the concave surface of the VAB. You should also see that a cap helix in gold lies within the bridge domain and would appear to partially block the lipid transfer. And at the right, you can see a bit of the bridge domain in light blue with a red line indicating how lipids would be expected to flow. And then you can see that the VAB domain in dark blue here blocks the bridge from accessing the membrane. So while the incellular studies by the Ticamilli group I referenced before show that VPS13C localization between the ER and late endosomes and lysosomes depends on VPS13's VAB domain, and the lysosomal marker GTPA is RAB7. It was not known whether VPS13C and RAB7 interact directly or how. So we undertook protein-protein interaction experiments, or really mini purifications, to obtain a better understanding of how VPS13C and RAB7 might interact. And in these experiments, we co-transfected mammalian cells with plasmids to express a strep tag version of RAB7 and a flag tag version of VPS13C, or mutant forms of VPS13C, passed the cell lysates over streptactin resin to retain RAB7 and any associated proteins, wash the resin to remove non-specifically bound proteins, and finally eluted RAB7 and any associated proteins. And then the eluted proteins were analyzed by SDS-Page and visualized either by Komatsu stain and or Western blotting. And we found that RAB7 specifically retains full-length VPS13C, although with relatively weak affinity because the retained VPS13C is representing only a small fraction of total overexpressed VPS13. The interaction depends on the presence of VPS13's VAB domain because a construct that lacks the VAB was not retained. And pay attention not to the blue arrow, which is endogenous VPS13, but to the black arrow, which is the VPS13 that lacks the VAB domain. And then in contrast, the VAB domain alone can be retained as detected by Western blotting. Removing other C-terminal modules, including the amphipathic helices or combinations of the WWE domain, the pH domain, and the amphipathic helices, did not abrogate VPS13C pull down, indicating that these parts of VPS13 are not required for this interaction. So Shimbo Wang in the Ticamilli lab then undertook experiments with VPS13C constructs and giant unilamellar vesicles. So using fluorescence microscopy, he showed that purified intact VPS13 interacts with GUVs that comprise both DOPC and deoleoglycerol lipids, where the latter are introducing packing defects. And that's in this column here, but not with GUVs that are lacking the deoleoglycerol lipids. constructs that lack the C terminus from the amphipathic helices in the ATG2C motif through the end did not associate with any of the GUVs, whereas a construct comprising only the amphipathic helices as present in cell extracts interacts with the dioleo-glycerol lipid containing GUVs. So these data reinforce Shimbo's previous findings that the amphipathic helices at the C-terminal end of VPS13C sense and promote association with lipid packing defective membranes, as you might find in damaged lysosomes. And then Dajili showed by flotation assay that VPS13C will associate with liposomes containing DOPE to introduce some packing defects where the when amphipathic helices are present. That's in the top. In the absence of the amphipathic helices in this ATG2C delta construct, membrane association is reduced by about 70%. Intriguingly, membrane association of this delta ATG2C construct can be rescued if Rab7 is present on the liposome. So this experiment was carried out with purified proteins, strongly supporting that the interaction is between VPS13C and Rab7 is direct and can take place in the context of membranes. So I haven't shown here last week's data that the rescue happens only in the presence of the VAB domain, which then supports a direct interaction between the VAB and Rab7. So our work on VPS13C in vitro has uncovered new aspects of its regulation and of VPS13 family protein regulation more broadly, pertaining to interactions both at the lipid donor and the lipid acceptor organelles. So one major outcome of this cryo-EM study was to identify an inactive conformation for VPS13C. We found that when VPS13C is in solution, interaction with the bridge domain C-terminal cap maintain the VAB domain in an arch over the end of the bridge domain. And then in this position, the VAB blocks the bridge domain from associating with membrane, and thereby it inhibits lipid transfer. We also identified a cap helix as partially blocking the end of the lipid transfer channel, presumably also inhibiting lipid flow to the membrane. So lipid transfer requires conformational rearrangements at the VPS13C terminal end, where the cap helix moves out of the lipid transfer channel and interactions between the cap and the VAB are loosened, allowing the VAB to move to the side of the bridge. The second major outcome of this study is identification of calmodulin as an interactor for a subset of human VPS13s that localize to contacts involving the ER as the lipid donor membrane. So as the binding site for calmodulin, VPS13 is at the N-terminal tip of its bridge, whose association with the ER is required to allow VPS13 to function in lipid extraction from this organelle. An attractive model is that calmodulin is mediating the connection between the bridge domain and the membrane. The presence of calmodulin, which is central to calcium signaling in VPS13 complexes, calls attention to an emerging role for calcium in the regulation of VPS13 activity. Such a notion is, of course, especially appealing because contact sites are hubs for calcium signaling. Then particularly relevant for VPS13C function at ER and lysosome contacts, calcium release by lysosomes is part of the stress response to their damage, and calmodulin could then confer calcium sensitivity to the ER localization and activity of VPS13s. Additionally, VPS13 likely is regulated by post-translational modifications at its N and C termini, and potentially flux could be regulated by modulating the width of the lipid transfer groove. So we've begun to understand how VPS13s could be regulated, but there's still very much to learn. So please hang on now for the next part of this talk. So I started this seminar off by telling you that BLTPs might cooperate with scrumbases, but I couldn't talk about scrumbases in the context of VPS13C because no scrumblase partners have been identified for it so far. So as promised before, in the remaining portion of the seminar, I'd like to describe the structure of another VPS13, VPS13A, in complex with a known scramblase interaction partner, XK, at the plasma membrane, in what we imagine could be prototypical for BLTP machinery as it assembles at the acceptor membrane. So loss of function mutations in VPS13A result in chorea acanthocytosis, which is a Huntington-like neurodegenerative condition, and a very similar condition called McLeod syndrome is due to loss of function mutations in XK, consistent with the functional partnership between the two proteins and their reported direct interaction in erythrocyte dynamics. Both VPS13A and XK are also required in T cells for the surface exposure of phosphatidylserine, suggesting a model according to which VPS13A can deliver phospholipids from the ER to the plasma membrane, where XK then can scramble them. VPS13A also localizes to other sites in the cell, such as contacts between the ER and the mitochondria, but its function there is less well investigated and less well understood. And so without further ado, here is the structure of VPS13A in complex with the scramblase XK as recently obtained by Boran Hu in our lab. So here at the left is a composite map put together from 4 reconstructions with average resolutions between 3.2 and 3.5 angstroms. And then at the right is the model by Boran. The structure, along with VPS13C that I just showed you, is the first one of an intact VPS13. It is the first near atomic resolution structure of a BLTP in complex with a bona fide scramblase, and it is the first indication of how an assembled BLTP machinery at the acceptor membrane might look. So XK is shown in pink here, and then the rest of what you see is VPS13, except for this purplish density near the N-terminal end, which represents the new VPS13 interactor, calmodulin, that Daji Li and colleagues in the Decamilli group have recently identified and which I've already discussed. If we were envisioning the VPS13A X complex bound to the plasma membrane, the extracellular face of the membrane would be here, and the cytosolic face would be here. The VPS13 bridge domain is again shown in light blue, and then the C-terminal end of VPS13 interacts with XK. As I mentioned before, all the VPS13s have a set of amphipathic helices near their C-terminus called the ATG2C motif, and these are not ordered in the reconstruction. In this reconstruction as well, probably because XK was extracted from the membrane in detergent, and again, there's no membrane present. You can also see the VAB in this kind of sickly yellow color, and also the pH domain in blue, and also present is the WWE module that's present in VPS13A and C. So as the map spins, you can see that in VPS13A, the VAB in this sickly yellow color, the pH domain in blue, and the WWE domain in green lie roughly in a plane that surrounds the C-terminal end of the VPS13 bridge domain, each positioned to interact with proteins at the acceptor membrane. In this case, only the pH domain of VPS13A engages a partner, which is XK. Indeed, the Decamilli group previously showed that the pH domain alone is sufficient to target VPS13A to XK and the plasma membrane. Most importantly, though, you can see here that the end of the bridge domain can access and is positioned to connect directly to membrane, meaning that lipids can be transferred directly from the bridge domain to the membrane, and then they're deposited close to XK so they can be readily scrambled. VPS13A's bridge domain is similar to that of VPS13C in its architecture, except that it comprises only 10 of these RBG repeats versus 13 in VPS13C, and it's, of course, much narrower. While we didn't expect for the lipids to be bound identically in all the hundreds of thousands of particles that we average for the reconstruction. In these higher resolution maps, we do see some density for acyl chains in the channel, indicating that there are a number of preferred binding sites. In the VPS13A XK structure, the VAB domain and the caps at either end of the bridge domain are less well resolved, reflecting that these regions are mobile. And indeed, you already saw the cryo-EM reconstruction of VPS13C by Jiajie Li, that the VAB can adopt another transfer inactive closed conformation where it arches over the bridge domain and blocks its access to membrane and so lipid transfer. The position of the VAB in the VPS13A XK structure is to the side of the bridge domain, allowing for the bridge domain to access the membrane. A variability analysis of the particles used in the reconstruction for the VPS13A XK complex shows the VAB in a continuum of positions which are more or slightly less permissive for lipid transfer. The same variability analysis shows that the opening of the VAB domain, which is kind of shown in the top row here, is coordinated with movements in the cap at the end of the bridge domain, where the lipid transfer channel appears to be partially obstructed when the VAB is closer to the lipid transfer non-permissive conformation and unobstructed when the VAB is in a more lipid transfer permissive conformation. We suspect that the opening up involves the alpha-fold predicted helices in the cap that are not visible in the structure, but are visible in the lipid transfer inactive structure of VPS13C that Dachi obtained that I showed you in the last slide and earlier in this talk. So the bridge domain of VPS13A does not come into contact with XK, and VPS13A binds to XK via a large interface of almost 2,500 square angstroms of occluded surface, and the interaction is exclusively via this pH domain of VPS13A. The binding site for the pH domain comprises a cytosol accessible pocket that's present in XK and all proteins in the XK family, and then a beta hairpin in XK. The pH domain associates with the beta hairpin, and then loops in the pH domain insert into the membrane into a pocket between interhelices 1 and 2 of XK, and also in this cytosol accessible pocket. Residues in this pocket are highly conserved, but the function of the pocket was unknown. So based on structural studies of XKR8 and XKR9, it had been proposed that peptide binding in this pocket might be inhibitory to scrambling, but this was never tested in a reconstituted system. We therefore used a well-established in vitro scrambling assay that was developed by the Menon group, where scramblase candidates are reconstituted into liposomes to assess whether binding of the pH domain of VPS13A might inhibit scrambling by XK. So we compared scrambling by XK alone and then by an XK pH domain fusion protein. We used an XK pH fusion construct to assure that all the XKs in this assay were bound to the pH domain and that there were no free XKs. We found that in our case, scrambling by XK and the pH domain bound version are similar, so that XK in the VPS13A XK complex can scramble. We therefore propose that the function of the cytosolic pocket in XK and related proteins might be for the recruitment of interacting partners like VPS13A for XK. At this point, I'm going to come clean and admit that despite now 4 experimental structures for XK family proteins, including XK in our cryo-EM reconstruction, the mechanism by which XK and related proteins scramble has been mysterious. So most characterized scramblases are thought to have polar channels through the greasy part of the membrane that allow the lipid head groups to slide through the membrane, kind of like a credit card sliding through a reader. And yet, No polar channel had been identified in any of the experimental structure, either by eye or by in silico molecular dynamics calculations. Consistent with the structures of other XK-related proteins and AlphaFold predictions, XK comprises two modules, each consisting of 4 transmembrane helices 1 through 4 in one of the modules and 5 through 8 in the other, as well as several interhelices, some of which that I've already mentioned before, like interhelices 1 and 2, which dip into but don't extend the whole distance to the lipid bilayer. To obtain insights as to how VPS13A and XK interact with membranes, we had begun a collaboration with the computational group of Stefano Vanni, which includes graduate student Christian Rocha Odoa. So when Christian examined scrambling by XK in the conformation observed in the cryo-EM reconstruction using coarse-grained simulations, he did not observe any lipid transfer events. So he then carried out all atom simulations and found that in the presence of VPS13A or just the pH domain, XK can adopt another conformation in which the transmembrane helices 1 and 4 have moved further apart. And then this motion opens up a polar pathway between transmembrane helices 1 and 4 with transmembrane helix 3 in the back of this pathway. And here you see the two conformations of XK, where XK is in a space-filling representation and its surface is colored by hydrophobicity. And you can appreciate that as transmembrane helices 1 and 4 move further apart, a polar channel opens up with transmembrane helix 3 at its back. And then in coarse-grained simulations, Christian now sees scrambling. Let's hear. So note that a mutational analysis of other XK proteins, notably XKR4 and 8, had identified residues along transmembrane helices 1, 4, and 3, which would be along the opened up channel as important for scrambling, supporting the hypothesis that the scrambling pathway might be between transmembrane helices 1 and 4. Okay. So the reconstruction shows that the end of the bridge domain appears to be positioned to interact directly with membrane, but we can't see from this reconstruction how this interface looks. We suspect that the portions of the cap that are mobile in the structure may be involved in the bridge domain interaction with the membrane, and we further suspect that the amphipathic helices in the so-called ATG2C motif that we also do not see could play a role in the interactions. The amphipathic helices in the ATG2C motif are, of course, of particular interest, as they're known to play important roles in the localization of both VPS13s and ATG2 family BLTPs, and because the amphipathic helices have the ability to bind and remodel membranes, which could eventually then play a role in lipid exchange. So at the right, please see a panel showing that these amphipathic helices mediate VPS13A membrane association in vitro. To better understand how the VPS13A XK complex interacts with membrane, the Vanni group further studied in silico how the complex might embed into membranes. And they first sought to understand how the bridge domain can be positioned close to the membrane so that lipids can be moved efficiently between the BLTP and the membrane. They found that the interaction between the pH domain and XK brings VPS13A's C-terminus proximal to the membrane, and then this facilitates the membrane insertion of the amphipathic helices in the ATG2C motif, and that this insertion is critical for close apposition. Further, the Vanni group found that the complex perturbs membranes locally. So we had noted that the micelle in the reconstruction is thinner on one side than the other, consistent with membrane thinning in a membrane. And then the Vanni group also saw such thinning primarily due to XK computationally. And indeed, scramblases often do thin membranes, and this is thought to facilitate scrambling. And a similar thinning, also near interhelices 1 and 2, was noted by the Accardi group for another member of the XK family, XKR4. Further, the Vanni group noted that the BPS13A XK complex induces local membrane curvature with contributions both by XK and the ATG2C motif. So membrane thinning and deformations perturb lipid packing, and they could facilitate lipid insertion as lipids are delivered from the BLTP bridge domain and then to the membrane. And then next, we assessed computationally whether lipid transfer between the bridge domain and the membrane is plausible. And by we, I mean Daniel Alvarez and Stefano Vanni. And here the lipids are in yellow with red head groups. The bridge domain is in teal, the amphipathic helices are in green, and then alpha-fold predicted helices in the cap are in orange. And we were initially very excited to see the transfer of a lipid. But to be frank, this is the transfer of a single lipid, and it seemed rather anemic. So arguably, the conditions in this experiment are not mimicking physiological conditions. So I think a major unanswered question regarding BLTP-mediated lipid transfer is what drives the directionality of lipid transfer. But everyone agrees that there has to be some kind of a driving force. So Daniel and Stefano modified the parameters to introduce a driving force. And perhaps most importantly, they stuffed the bridge domain with lipids, 93 versus 54 or 75 as in the previous picture. And then also they applied membrane tension. This is essentially to mimic lipid synthesis in the endoplasmic reticulum. And then they also applied membrane tension to the acceptor membrane. So membrane tension is low in the endoplasmic reticulum, and it's higher in other compartments. And the current thought is that the difference in membrane tension could be a driving force for directional flow. And then you can see what happens when we add this driving flow. Now we are going to get robust lipid transfer. So we see now robust transfer of about 27 lipids in kind of all at once. So just to rehash, we saw robust lipid transfer when we introduced driving forces into this in silico system. Although the nature of the driving force in real life is not established, the experiment by Daniel and Stefano support the hypothesis that differences in membrane tension between the ER and other organelles, including the effects of asymmetric lipid synthesis, could contribute to this force. So our model so far for lipid delivery by the VPS13A XK complex is that the BLTP bridge domain connects to membranes and delivers lipids directly to it. The interaction between XK and the pH domain anchors the bridge domain at the membrane and orients it, and along with amphipathic helices in the ATG2C motif, pins the bridge domain close to the membrane. Lipids are delivered to the membrane close to the scramblase, which enables bilayer expansion. Membrane deformations may facilitate lipid delivery, and then a driving force is required for robust lipid delivery. The current view is that differences in membrane tension, including by asymmetric lipid synthesis between the donor and the acceptor membranes, could provide this force. BPS13A may function similarly at other sites, using other of its adapter domains that might interact with other scramblases or integral membrane components. Other VPS13s have similar domain architectures and likely function by the same mechanisms. The ATG2 proteins are pared down versions of VPS13, comprising just the bridge domain and the amphipathic helices, and they likely deliver lipids by the same general mechanism as the VPS13s, except that additional adapter modules are provided by exogenous proteins. oops. And then the proteins or adapter modules that mediate membrane attachment by the bridge domains of other BLTPs still remain to be identified, but most likely the same general mechanisms are at work. So several months ago, a paper came out showing how BLTP1 accomplishes lipid uptake, which is via an integral membrane vestibule that would appear to facilitate lipid egress from the donor membrane and into the bridge domain. Our results now allow us to describe the whole lipid transfer process, as they provide insights as to how lipid delivery to the acceptor membrane might be accomplished. So lipid delivery is not to an integral membrane protein vestibule, but directly from the bridge domain of the BLTP to the acceptor membrane. We think it might be facilitated by membrane perturbations and deformations introduced by the BLTP and the scramblase, and rates are almost certainly affected by driving forces. Our hypothesis is that asymmetry in lipid biosynthesis, which takes place in the cytosolic leaflet of the ER, may be critical, but that other sources of membrane tension could also be important. So finally, I would like to thank my coworkers, and in particular, Bodan Hu, who's primarily responsible for the structural studies of VPS13A and XK, and Daji Li, who's responsible for structural studies of VPS13C and calmodulin. I am ever so grateful to Christian Rocher-Rohr, Valentin Guyard, and Dani Alvarez and Stefano Vanni's group for their ever so cool computational analysis and studies. And I would like to acknowledge Pietro De Camilli and his group, especially Shimbo Wang and Hongyan Hao, for their help with BPS13C study, and in general for continuing discussions and collaborations. And I'm happy to take questions.
Thank you, Karen. That was really beautiful, super fascinating what you shared with us today. To give people the time to submit questions to the Q&A or to raise their hand, we can maybe get started with Jim.
Karen, fascinating progress. I have a question really around the role of the pH domain, given how extensive the contacts are. It seems to be an integral part of the complex that you showed in the simulations. It seems a little surprising it's not necessary for the in vitro lipid transfer. I'm wondering if that has to do with, perhaps what's the two-dimensional concentration and how that compares to the cellular level. So perhaps if it's overly concentrated in the in vitro model, then the contribution disappears. So one question is about that possibility, and the other is what's going on with ATG2, which doesn't have a pH domain. What substitutes for that?
So, Jim, your first question is that whether the pH domain has to do with scrambling, or does the pH domain have to do with?
It has to do with both. You showed in the in vitro
Yes.
reconstitution it's not required for the
So yeah,
scrambling. So you're suggesting it's only involved in targeting, but I'm wondering how sure you can be about that because of things like dilution effects maybe could become important at some other concentration, or perhaps not really sure.
So I mean, the in vitro scrambling assay, as well as the in silico simulations, have to be taken always with a grain of salt. We try to mimic the composition of the plasma membrane, but you know that it's hard to mimic an asymmetric membrane in liposome assays. So maybe we didn't get that right, and there could be differences depending if the pH domain is bound or not, like in a real life scenario. And then in silico, in silico computations, the pH domain seems to push the pH, the scramblase into a more active conformation, which doesn't take place without the pH domain. But again, it's very hard to figure out what is how much it's neither one of these are real case scenarios. So probably one should try to redo some of these experiments in different membrane contexts, or maybe in cells. I'm not sure. What was the second question?
So ATG2, which doesn't have a pH domain,
Yeah.
what substitutes for the
Well,
missing pH domain in the ATG9 interaction, which is the counterpart of the XK?
so there are two papers that maintain that ATG2 feeds lipids directly to ATG9. These two structures are, well, in my view, both of them suggest that there's a very good tethering between ATG2 and ATG9. And so the ATG2, ATG9 interaction may in part help to pin ATG2 close to the membrane. I would imagine that the WIPI also probably interacts with PI3P and has a role in pinning ATG2 close to the membrane. The parts that are conserved or that are important are, of course, these amphipathic helices, which probably enable a very close apposition of the bridge-like domain and the membrane. So those are present in all of the VPS13s and the ATG2s. I think there's also these alpha helices in the very end, the very C-terminal tip of VPS13, predicted by AlphaFold, and those probably also come undone, I would imagine, in ATG2 as it's kind of interacting with membrane. But I would, rather than the pH domain, I think in all of these proteins you can swap out the adapter domains or the tethering domains.
Thank you. And the next question is from Chao Li, asking basically two questions. I'll try to summarize them. What are the type of lipids that are being transported by the VPS13s? Are they also neutral lipids or only the phospholipids? And I think related to that is the second question. What is sort of the receptors on the ER? Do they associate with like the lipid synthesizing machinery at the ER site?
Okay, so this is a very good question to which we do not know the answer. So when we initially overexpressed VPS13 proteins or also ATG2 proteins and analyzed the lipids that they co-purify with, they were mostly glycerophospholipids in the same ratio as they occur in cells that we were purifying the proteins from. So that would suggest maybe that these proteins intrinsically don't really have a preference except for what's there. That doesn't mean that that they're not transporting more specifically in cells, because if it's true that they are interacting with integral membrane proteins in both the donor and the acceptor membranes, you could envision, for example, that at the donor membrane, the integral membrane protein might control access to the channel. It's also possible that at the donor membrane, these complexes could be assembled in patches of membrane that have a more like in like a micro composition that's enriched in one glycerophospholipid or another. So we don't know. For BLTP2, I believe it's been speculated that it's involved in transferring PE because it functions in providing, at least in some organisms, PE to the plasma membrane to make the plasma membrane more fluid. So, but does that mean that it's just sitting in a place where there's more PE at the N-terminal end, or does that mean that it's specifically transporting PE? I think we don't know that.
Okay, thank you. Next question is from Emily Millard. She said, thank you for the talk. It's a bit of an extensive question, so I'll try to summarize it for you. Although VPS13A and VPS13C share a similar modular organization, the positioning of their pH and VAP domains differs in the way that the pH domain of VPS13 binds PI3P and PI5P, and the yeast homolog also does that. So This raises the possibility that the pH domain of VPS13C may similarly interact with phosphoinositides, providing an alternative mechanism for lysosomal targeting. So basically, the question is that in absence of RAP7, is there a possibility that VPS13C, through its phosphoinositide sensing, would still be recruited, whether that would be a plausible alternative?
So I was not aware that the pH domains in either of these proteins interact with phosphoinositides. So this is not, and I'm pretty sure that you need RAP7 for the localization to the lysosome. But I haven't looked too closely at that. I mean, given that myself. In the VPS13A structure with XK, the pH domain is far removed from the plasma membrane, or too far removed for it to be interacting with phosphoinositides. And also we didn't find a basic patch as you would expect for PIP association. And I haven't looked at VPS13's C's pH domain in that much detail.
Okay, thank you. And then maybe one last question from my side before we close the session. During your talk, you mentioned that RAP7 is critical for its recruitment, and we know that RAP7 gets phosphorylated. And then you also mentioned that the VPS13C itself might be phosphorylated, and we know that, for instance, LRRK2 gets activated during LOMI treatment. Is there any link between RAP7 phosphorylation and VPS13 recruitment, or LRRK2 and VPS phosphorylation?
I know for sure that the Decamilli group has tested whether RAP7 phosphorylation makes a difference and RAP7 phosphorylation doesn't make a difference. And I don't know anything about, I did check, but I forgot the answer whether LRRK2 is reported as phosphorylating VPS13C. So I don't know, but yeah, I can't answer this question.
That's fine. Thank you very much. It was really fantastic. I think all that's left to do, since we're a little bit over time, is to thank you, Karen, for this fantastic talk, and to invite everyone for our next speaker, which is going to be on the 11th of December. We have Mika Rape, and it promises to be another really fantastic session. So I hope to see all of you there again. Thank you, and have a nice day and evening.
Bye.
Thank you, Karen, for your time.
Thank you.